you have passed your exams. You are quite good in your content, right? So you only need to learn how to link topics together and be better able to answer the questions that appear in your exams. So today we are looking at this topic on molecular genetics and genetic engineering. So as you can see, we are actually linking two topics together, molecular genetics and genetic engineering. So how do you prepare yourself well for an exam? Make sure that you can do well in that paper. There are a few things you need to consider, right? So if you want to do well in the exam, firstly you must know what to study. Right now, you must know what to study. So for this particular topic on molecular genetics and genetic engineering, what is required in the syllabus that you need to learn up? First thing you need to know. The content, so you go and learn up those things. Now, the second thing is, after you know your content, the second thing is, you should also try to also find out what were the questions that have come out before in the past 10 to 20 years ago. As many years as possible, that syllabus, right? So the TYS is a very good resource. So we will be looking through the TYS at what kind of questions come out before. Because at the back of your mind, if you know what questions come out before, when you do your revision, you will zoom in and focus on those areas. And you will not miss out what is important, right? So if you know what to study, you know what are the questions that are likely to come up. The third thing is, are you able to answer the questions? Do you have the technique to answer those questions to gain as many marks as possible? So these are the three important things that we hope to cover in this session. So for this chapter on molecular genetics and genetic engineering, these are the things you need to know. You need to know the structure of DNA. You need to know the role of DNA in protein synthesis. You need to know genes. Okay, what has genes got to do with protein synthesis and structure of DNA? Right? So, in other words, you need to you need to know what are the learning outcomes. So this is very important. So I'm going to run through the learning outcomes. I'm going to ask you questions to see whether you really have content for all this before we move on. Okay, firstly, students are supposed to be able to outline the relationship between DNA, genes, and chromosomes. Do you know the relationship between DNA, genes, and chromosomes? Like if you're asked what is DNA, do you know what DNA goes? Huh? Where do you find DNA?
In my cell.
Huh? In the cell? Which part of the cell?
My cell.
Huh? Which part of the cell? Let's quickly answer so that the nucleus, right? Now, do you know the relationship between genes and DNA? You know or don't know? Check with your friend. You don't know, I have to explain. If you know, I will just move on. Because you're supposed to know, Do you know the relationship between genes and DNA? Yes or no? Don't know Okay, let's say this is a DNA molecule, right? Very, very long molecule. Where are the genes?
On it.
The genes are on the DNA molecule. This part is a gene. Maybe somewhere further down. Another part is a gene. Another part is a gene, right? So you need to know that genes are actually segments of the DNA, which is consisting of a sequence of nucleotides. Then what about chromosome? How is DNA related with chromosome? Do you know? If you know, we move on. Don't waste time. Do you know what's the relationship between DNA and chromosome? Which is bigger? Chromosome. Then how are they different? See, the DNA is a very, very long molecule, right? Does it exist as a long, straight molecule? Oh. It gets twisted into a double helix. Does it just remain as a double helix? No, because it's too long. So what happened to it? It will coil around, wrap itself around proteins. So when it wraps around proteins, it forms chromatin threads. What about chromosome? What about chromosome? So chromatin thread is the long DNA wrapped around proteins, you get chromatin thread. Then how is this related to chromosome? Chromatin thread is still long. You need to make it even shorter, right? Especially during cell division, these chromatin threads will become even shorter. How do they become shorter? They condense by coiling upon themselves to become shorter to form a chromosome. You know, I wish I had a space to draw to show you this. How to draw like this?
So you can move the board.
Then you see capture as I say. Okay, so if this is a very, very long double-stranded DNA, these are protein molecules. The DNA coils around the protein, become shorter, right? This is a chromatin thread. The chromatin thread, it is the form in which DNA exists inside the cell. That's chromatin thread. But when the cell is about to divide, this chromatin thread will even condense further. So this one will coil upon itself, become even shorter, you get a chromosome. All this you should know. So there's a relationship. Next, state the structure of DNA in terms of bases, sugar and phosphate groups found in each of the nucleotides. This one, do you know? I don't think I want to waste time explaining because you're supposed to know. Okay, so are you able to describe the structure of DNA in terms of bases, sugars and phosphate groups?
Yes.
How would you describe it? Yeah? How would you describe? DNA and also nucleotide. What is a nucleotide? What does a nucleotide consist of? Nitrogen containing base, phosphate group, and a deoxyribose sugar like this, right? So, deoxyribose sugar, a phosphate group attached to a base. This is a nucleotide, correct? So DNA is made up of many nucleotides linked together to form one long polynucleotide. Then you have two polynucleotides which are linked by complementary base pairing. Do you know the rule of complementary base pairing? Adenine with thymine, cytosine with guanine. DNA is used to carry the genetic code which is used to synthesize specific polypeptides. Details of transcription, translation not required. Do you know how DNA carries a code to synthesize specific polypeptides?
No.
Huh? No, this I'm not sure. Need to explain this, is it? Is this clear? Need to explain. Okay, if this is not, so I'll just go over things that you are not so clear about. Okay, DNA, all this I don't have to go through, all this you know, right? Double-stranded molecule. You know what's a gene? A gene is a segment of the DNA. So let's say if this part of the DNA carries a code to synthesize a protein, then this is a gene. If this part of the DNA has no code, it doesn't code for a polypeptide, then this part is not a gene, and this part is a gene. Okay? So therefore, the long DNA molecule is divided into segments of gene and junk DNA. We go on to protein synthesis. Okay, so protein synthesis, we need to know what is transcription and translation though. This one is a gene. Why is this segment called a gene? Because it carries a code which will tell the cell what polypeptide to make. It codes for the synthesis of polypeptide. The code is only written on one of the two strands. Okay, let's say the code, this is DNA is double-stranded, right? So So if the code is written on this strand, but not this one, then this strand is called a template. So the template carries the code to tell your cell what polypeptide to make. Okay, next. How is this template, how does it code for the polypeptide? Now the code is written in the form of a triplet code. That means this is the template, right, that codes for the polypeptide. Triplet code means every 3 bases, TAC, code for 1 amino acid. The next 3 bases code for another amino acid. The third 3 bases code for another amino acid. So when this template is decoded, TAC will be decoded to give you this amino acid. TAT represents this amino acid. So during protein synthesis, these amino acids will line up according to the order in which the bases appear in the template. And then they're linked up by peptide bonds to form a polypeptide. Okay, so therefore, the sequence of bases on the DNA is very important because it determines the sequence of amino acid, right? The sequence of bases determine the sequence of amino acid. The sequence of amino acid specifies what the polypeptide is. Different polypeptides have different function. So if you have a change in any of these bases, you will change the amino acid and you get a different polypeptide which will produce a different function. Okay, this one you know. So every 3 bases code for 1 amino acid. Okay. This one you know, I think. Okay, so how is protein being synthesized? This is the cell. Inside the cell, you have the nucleus. Inside the nucleus, you have the DNA. Right? On the DNA are genes. So let's say this part, there is a gene here, this gene. Now the gene is double-stranded, right? So the code is only written on one strand. So what do we call this strand in which the code is written? Template, right? So the template carries the code for the polypeptide that is supposed to be made. Now the code is inside the nucleus, but protein synthesis occurs outside the nucleus. You know, I bring you back to this. Do you remember this diagram? Do you remember this diagram in Sec 3? So I'm trying to link for you. You know, in Sec 3 you learn that inside a cell, okay, inside a cell, you have endoplasmic reticulum, right? On the endoplasmic reticulum, the rough endoplasmic reticulum are the ribosomes, correct? What is the function of ribosome? Synthesize protein. How do the ribosomes know what proteins to make? It depends on the instruction given by the nucleus, right? So this is where protein is made at the ribosomes. But how do the ribosomes know what proteins to make? The instruction is carried inside the nucleus in the genes on the DNA. So if the instruction is inside the DNA, which is inside the nucleus, and the factories that make the proteins are outside, then the instruction here must be relayed outside, right? Okay, so there must be a way to convey the instruction here out to the ribosomes, to tell the ribosomes what proteins to make. So after the proteins are made, what happens to the proteins? The proteins will be pinched off in vesicles, which will then be carried to the, what is this thing? Golgi apparatus. At the Golgi apparatus, these polypeptides that are made will be modified, right? Repackaged into proteins that are needed by the body. They are packed into vesicles, which are pinched off, and then they leave the cell. So this is proteins. This is what we learn in Sec 3 lah. So what we are learning in Sec 4 that relates to this is that inside the nucleus you have the genes on the DNA. The gene carry the instruction as to what protein to make. Okay, so this instruction must be relayed to the ribosome so that they will make the right proteins. So how to relay the instruction there? You have to copy the code on the gene into another molecule that is able to leave the tiny pores on the membrane of the nucleus. Okay, the DNA is too big to come out. Right, so I go back to the other slide. See, the DNA inside is a very, very long molecule. DNA does not leave the nucleus, cannot pass through. So how to get this code to be, the information here, to be carried out to the ribosome? This is a ribosome. So what happened is, first you have to copy this code into another molecule. You know the word scribe means copy, right? Take notes, right? So transcription means that the code on the template is copied onto another molecule called the mRNA. So this mRNA carries the code, and then it is small enough to leave the nucleus and attach itself to the ribosome at the endoplasmic reticulum. The ribosome will then decode. Right, so this is transcription. It happens inside the nucleus. Transcription means copying the genetic code on the template into another molecule. What do I call this molecule, girls? mRNA, right? Okay, so transcription means you copy the message on the DNA template into the messenger RNA. It occurs inside the nucleus. So how to copy? So that part of the DNA which carries the gene must unwind, remember? So if this is the template, then the code here will be copied onto the mRNA by complementary base pairing. So if it's T, you copy as A. If it's A, you should copy as T, right? But mRNA does not have thymine. It replaces it with uracil, U. So you copy, copy, copy until the end. So this is the mRNA. It is small enough, it can leave the nucleus. Okay, so you will leave the nucleus. You see, the mRNA will leave the nucleus, go into the cytoplasm. Okay, so once it leaves the nucleus, it will go into the cytoplasm to where the ribosomes are, either the free-floating ribosomes or those that are attached to the endoplasmic reticulum. And at these places, the protein is being made. Okay, so how is it done? So what happened after transcription is that the mRNA leaves the nucleus, go into the cytoplasm, attaches itself to a ribosome inside the cytoplasm. At the ribosome, this mRNA, the code here is decoded. You try to interpret the code. Okay, you try to translate the information here into the language of protein. So how does it happen? So this ribosome will then move along the mRNA. As it moves, it will read the code AUG. Now AUG is a triplet code. It stands for a certain amino acid. So as the ribosome moves, it translates the message on the mRNA into a protein molecule. So if it's CCC, it is the enzyme proline. It is the amino acid proline, right? If it's AAA, then it stands for the amino acid lysine, right? If it's CUA, it stands for the amino acid leucine. So you see each amino acid may be coded by more than one code. So that's how they code. Okay, so as the ribosome moves, the first three bases form a triplet code. It stands for this amino acid. Then it reads the next triplet code which code for this amino acid. Then it reads the next triplet code which code for this amino acid. So as it moves, it will translate. Amino acid by amino acid will be linked up to form the polypeptide. Then the polypeptide is synthesis. It is synthesized, linked up. This is translation. It occurs inside the cytoplasm. The details as you see in the synthesis is not required. You don't need to know great details. Okay? So what do you need to know if you don't need to know great details? You need to know what's the meaning of transcription. What's the meaning of a template? How is the template copied into the mRNA? What happened to mRNA? Right? How does translation take place? It takes place at the ribosome where every 3 bases code for 1 amino acid. The amino acids are linked up according to the sequence of codes here. You get a polypeptide. Okay, so transcription, translation. You get your polypeptide. By the way, where do these amino acids come from? We are saying that at the ribosomes, the amino acids are all linked up to form a polypeptide. Where do the amino acids come from? Where do the amino acids in the cell come from? How come your cells have so many amino acids? Where? This one you know, right? How come your cells have so many amino acids and they are available to make the polypeptide? Where do the amino acids come from? Digestion of the proteins you eat. So you see, you link digestion here, right? Every day you eat meat, you eat protein. After digestion, what do you get? Protein is digested to amino acid. The amino acid is absorbed by the blood and then is carried to your cells, diffused into the cells. All the amino acids that diffuse into the cells are waiting in the cell for instructions to make the polypeptides, the different polypeptides.
You know, like after the ribosome moves along the mRNA, what happens to the mRNA?
The mRNA can attach to another ribosome and repeat it. Because you don't only make one protein, you make again and again, right?
For each one, right, can take a long time.
Yeah, the mRNA can make a lot of times. Unless the protein is no longer needed, then mRNA may be destroyed.
What happens to the polypeptide?
The polypeptide, what happened to the polypeptide that's made those? The polypeptide will find its, will be enclosed inside a vesicle. Oh. Right now, it will be enclosed. So the polypeptide that's made is enclosed in a vesicle that's pinched off, right? Okay, so those proteins will then be carried by the vesicle to the Golgi apparatus, where these proteins are being modified into secretory proteins. So you have vesicles carrying the secretory proteins that are secreted out of the cell. Okay, so you have to link digestion of proteins, amino acids. Amino acids are absorbed into the cell, used for protein synthesis. How are the proteins made? What proteins are made? Depends on the instructions here. Depends on what genes are present there, and what mRNA has been sent out to the ribosomes. Okay, so this is called making linkages. So this is what you have learned. So now you pull everything together. So this is what you have asked me. Vesicles transport the substances within the cells, right? So they are pinched off. So all the proteins that are made are pinched off, and then they will float across to the Golgi apparatus, fuse here, and then modify and pinch off again. You get the secretory vesicles with the modified proteins to be secreted out of the cells. Okay, so they are pinched off. Okay, so that's the protein synthesis part. Is this part not clear? By now clarified already. Okay, next. State that each gene is a sequence of nucleotides as part of the DNA. This one you should know. Now the other thing about genetic engineering is this. For genetic engineering, what do you need to know that will be tested? Now you need to explain that genes can be transferred between cells, right? You can actually, scientists can now transfer a gene from one organism to another, and that process is called genetic engineering. So the genes can be transferred from one organism to another organism of the same species or even of different species. For example, from the human DNA to the bacterium, different species, right? Explain how a gene that controls the production of human insulin can be inserted into bacterial DNA to produce human insulin in medical biotechnology. This one is very important. We need to know how scientists can genetically engineer bacteria to make insulin. Do you know the process? You got the human DNA with the insulin gene, right? Then what do? You use restriction enzyme to cut out that human insulin gene from the DNA, you're supposed to paste it on a plasmid, right? So the next thing is to prepare the plasmid. Where do you get the plasmid? From other bacteria. So you have to isolate the plasmid from the bacteria, and then you use the same restriction enzyme to cut the plasmid, remember? So it produces sticky ends. So you put the cut plasmid and you put the insulin gene with sticky ends together. How do you glue it together? Using another enzyme called DNA ligase to seal it. So now you get a recombinant DNA. Okay, remember what you do with the recombinant DNA? The plasmid with the insulin gene, you put it into the bacteria. How? How do you put it in? How do you insert it in? Shock the bacteria. How? Heat treatment or electric shock. So the pores on the membrane open, and then this recombinant DNA enter carrying the insulin gene. So once the bacteria have the insulin gene, what do? They respond to the instruction of the gene, right? The gene will actually code for insulin. So it makes the bacteria produce insulin. So if you culture the bacteria in large quantity, you're going to get a lot of insulin, which can be extracted, purified, and used, as in the medical industry. Okay, so this one, social and ethical implications of genetic engineering with reference to a name example. So what can you think of? What are social and ethical implications? For example, the onco mouse. You know onco mouse? Yeah. So scientists have inserted the onco gene into the mouse. What does this gene make the mouse do? Produce tumors. The mouse will develop cancer and a lot of tumors. Why do you want to make the mouse produce tumors? So cruel. So that you can use this onco mouse for experiment to test the effectiveness of different drugs on the tumor. So if it found to be effective, then you can try on human beings. You want to try on human beings first, right? So there are social and ethical implications, So what does it mean? Some people think it's very cruel. It's not right to be so cruel to animals. Make them produce tumors, right? But then on the other hand, if you do this is one of the ways in which you can advance science and find maybe cures for cancer, effective drugs to treat cancer. So you have to weigh these two. Some people are against it. It's not fair to the animal. Some people say, never mind, sacrifice some animal to save a lot of people. So these are social, ethical implications. What else? Maybe even introducing the gene BT, like that, in the BT corn, right? There's another one. You introduce the gene into potatoes. Alright? So that it will make the crop resistant to aphids, right? Remember that one? Yeah? Okay, so the gene from some bean plants is inserted into some other crop plants, potato. Okay, so as to make these potato plants that are genetically modified able to be protected against aphids, so you have a bigger crop of potatoes, right? But what is the problem? Some people are allergic. Okay, allergic to the protein lectin that's produced by this gene, So they avoid beans or nuts, but they don't know that they have to avoid potatoes. After eating the GM food, what happens? They develop all kinds of allergic reactions. So there are implications again. So there are quite a number of examples you can read over. Okay, so this is the content you need to know. I run through. So if anything you're not very sure, you better go home and read up to make sure that you are okay. Next, we want to look at questions that came out over the last at least 10 years, okay? Okay, firstly, let's quickly run down to this. What is the correct arrangement of components in the nucleotide? Easy? Okay, right? See, the diagram shows process of genetically modifying bacteria to produce human insulin. Okay, it shows you these processes. Which stage use a restriction enzyme? Can answer? Can, right? So we won't go through. A gene contains 900 phosphate groups. How many bases will it contain? 900 phosphate group. How many? How many bases will it contain? A gene contains 900 of these. So how many bases? We say 900 lah. Correct? Right now? Because every time you get one phosphate group, it's linked to one base. So you have 900 phosphate group, you must have 900 bases. Okay, you see here this one? Stages in the transfer of gene from one species to another species are listed. So what are you required to know? You need to know the steps involving genetic engineering to introduce a gene into bacteria. So do you know the steps? So okay, you need to know this. Same thing, do you know the steps? Repeated question, right? Which one of these is part of the other three? So you need to know the meaning of chromosome, gamete, gene, nucleus. So how to answer this? Which one of these is a part of the other three? Which is a part of the other three? Oh. Is a gene a part of a gamete? In the gamete, do you find gene? Yes, what? In the chromosome, do you find gene? Yes. Inside the nucleus, do you find gene? Yes. So the gene is a part of all this. A chromosome is not part of a gamete. A chromosome is not part of a gene. Okay? So this kind of relationship you need to know. Which statement applies only to a gene? So again, you need to know what actually a gene does. So which one applies only to a gene? Which one? Can a gene be copied during cell division? Replication of DNA, what do you do? Before cell division, what do? You have to copy the DNA, right? You also must copy the gene. So the gene can be copied. Actually, it's before cell division. The gene control multiple characteristics. A gene, no, right? A gene control one characteristic. It calls for a single polypeptide. Yes. Wait, look like this one also can, this one can. It is composed of 4 bases. Which statement applies only? Only to a gene. Apply only to a gene. So this what they highlight. Which one? So during cell division, it's not only the gene that's copied, it's the whole DNA that's copied. So maybe this one can rule out only to a gene. It can control multiple characteristics. No. A gene code for a single polypeptide. This one only apply to a gene, right? One gene, one polypeptide. A gene composed of 4 bases is true, but it doesn't only apply to a gene. The whole DNA is composed of 4 bases. Understand? So for this type of question, I'm going to be very careful. Only to a gene. Okay, let's go on. This question is about synthesis of human insulin again, you see. So you need to know what are the enzymes that are needed and which stage. So you see how important this process is, right? So many questions, Okay, 100 nucleotides are mapped on a single strand of DNA. How many thymine nucleotides will there be on the strand of DNA that's complementary to this strand? One single strand of DNA. One single strand of DNA, you have how many thymine on the complementary strand? So this is a complementary strand. Now, if you find thymine on the complementary strand, that means there must be alanine on the original, the other strand, right? So, how many thymine will you find on the complementary strand? Depends on how many adenine you can find on this strand, right? So, adenine you have 22. So you have 22 and adenine here, I should have 22 thymine on the complementary strand. So 22. Okay, correct? So this tests you on complementary base pairing. Which statement about DNA is correct? I think we have gone through this before. You see the diagram? This is quite easy lah. Complementary base pairing also. This type of diagram come out, right? Don't have time to go. So you are supposed to identify what is W, what is X, what is G, you know? Yeah. So this is what? This is a sugar, right? This is a phosphate, this is a base. These are the bases. Another question on insulin production. Here they test you on the enzymes. So you need to know this process and enzymes. Which part represent a nucleotide? Okay, the structure of the DNA. What is this? A nucleotide consists of this one, phosphate, sugar, base. So where's the nucleotide? Where's the nucleotide? Yeah, phosphate, sugar, base. What describes the genes in a transgenic organism? Come on, this one. What describes the genes in a transgenic organism? Which do you think is the correct answer? Huh? Genes in a transgenic organism, is it derived from two organisms? The genes come from two organisms, right? It can be from more even. So this is possible. The genes in a transgenic organism have mutated naturally. It's not about mutation. It's inherited from two individuals. Inherited is wrong. Right? In transgenic organism, you don't inherit. It's not passed down from your parents as inherited. It is introduced from another organism. So this word with problem, inherited, no. Modified by radiation, no. The genes in the trans, it's not because it's modified. If it's modified by radiation, that is mutation. In a transgenic organism, it's not because it has mutated. That's why it's transgenic. Okay, this one's straightforward. What describes a transgenic plant? So what do you think is the good answer here? Transgenic plant. Is it a plant that's artificially inserted with genes from a plant of a different species? Possible, right? Is it a plant in which some of the DNA has been removed? No. Transgenic means you acquire some foreign gene. A plant which has acquired a new gene by mutation is not by mutation. A plant which has been bred by selection and controlled breeding? No, this is selection. So this is not transgenic. So answer is this. But it's also not the best answer because a plant artificially inserted genes from a plant of a different species. It could be from the same species. It could be maybe not even from a plant. It could be from a bacteria, right? But this is still the best answer out of 4 options. So sometimes you've got to study the options very carefully before ruling out, okay? So always possible, possible, possible. Then read again, read the question again. You may have missed out something. This is homologous chromosome. What statement describes a gene sequence of nucleotides, right? Transgenic maize plants. This one maybe you try. Produce chemical that kills insects feeding on it. Which implications of this situation are correct? Transgenic maize plants. Produce chemicals that kills insects. The insects beneficial to other plants may also be killed. Is it possible? Yes. Okay, so you want to kill the butterfly, but in the end you also kill the bee or the dragonfly. Food chains can be disrupted. Yes, the moment you kill some organism, you are going to disrupt the whole balance in the ecosystem. Yes, less weed killer is used by farmers. Yes or no? This chemical kill insects, not kill weeds. If you say less insecticide is used, yes. Less weed killer, not necessary, right? Because the insecticide doesn't kill weeds, so you may still have to use the same amount of weed killer. So read carefully. Now we look at what has come out in the past years. This is very interesting. So when you study, you know what to focus on. Outline the relationship between DNA, genes, and chromosome? What is DNA? What's gene? What's chromosome? Gone through, right? State the significance of the order of bases in a DNA molecule. What's the answer to this? What's the importance of the order of bases? If you change the order, what does it mean? Huh? What is the significance of the order of bases? The order of bases will determine the order of amino acids in the polypeptide. Am I right? The order of bases determines the order of amino acids in the polypeptide. Right? The order of amino acids will determine the function of the polypeptide. Correct? So this is the importance, because the order of bases determine the order of amino acids. The order of amino acids determine the type of polypeptide, and the type of polypeptide determines the function. And so if you change a base, you're going to change the polypeptide, you're going to get a different function. Instead of functioning as an enzyme, it will not. So the person doesn't produce the enzyme. Instead of getting normal hemoglobin, they get defective hemoglobin, just because one base is changed. Understand? So that's how important the order of bases are. Okay, next one. Describe how bacteria can be used to produce human insulin. This one, take note, is many marks, 5 marks, okay, came out in 2009. In other words, please go home and learn now. Describe the whole process of how you genetically modify bacteria to produce insulin. Okay? Next one. Describe the structure and function of DNA. Again, structure of DNA. So you must talk about the base pairing, you must talk about polynucleotides, you must talk about the nucleotides. Learn up this, Next one. Describe what is meant by a transgenic organism. This one, 3 marks. What should you write? What is meant by transgenic organism? Transgenic. How do you determine whether the organism is transgenic? If it's transgenic, that means it has acquired a foreign gene, right? Okay, so a transgenic organism is one that's acquired a foreign gene. Will that give you 3 marks? So next, what else can you write? What else can you write? So you may elaborate by saying, where does this foreign gene come from? This could be another point, right? The foreign gene may come from the same species of organism or from a different species of organism, right? So that could be the second point. What else to get 3 marks? So they have acquired a foreign gene. The foreign gene can be from the same species or from a different species. Then what else? So what if they have acquired a foreign gene? What will happen? How will they be different? The transgenic organism can then express the characteristic controlled by the gene. How? By producing the protein coded by the gene. So if you see there are many marks, try to elaborate and expand on your answer. Then you'll probably be able to get more marks. Don't just say, oh, I only learned transgenic organism has a foreign gene, full stop. What else are 3 marks? Where does the foreign gene come from? After it has a foreign gene, how is it different? Right? It can express a characteristic. How is it that it can express a characteristic? Because it produces a protein that is coded. Can I repeat? I'll repeat 2 times, 3 times. I watched the video. Okay, next one. Suggest 3 reasons for the development of transgenic organism. This is very general. There are many reasons, right? It is, for example, oncomouse. What is the purpose of producing? Yeah, so that they can be used for cancer research, right? To test on the effectiveness of different drugs in the treatment of cancer. Okay. Another reason. What else? Transgenic plants. Why are transgenic plants produced? Why do you want to produce golden rice, for example? You may increase the nutritional value of the crop. Why do you produce BT corn? You can protect your crop from pests, insecticides, and so on, pests and insects and so on, right? What else? What other transgenic organisms can you think of and why are they produced? What else? Insulin. Bacteria that produce insulin. What's the purpose? You can lower the cost of production of drugs, right? For example, insulin. The cost of producing insulin will drop to a fraction of what it used to be. It used to be like $100, now it's only $1. Big drop. Okay, so there are many reasons. So for this, you could think of examples. Again, girls, not many years later, describe the structure of DNA. How many times it appeared? Right? So DNA, how would you describe DNA? Consists of 2 polynucleotide chains that are what? Antiparallel. Okay? Each polynucleotide chain consists of nucleotides, right? So you can talk about the nucleotide is made out of deoxyribose sugar, a phosphate, and nitrogen containing base. Okay, then you talk about the base now. There are 3 bases, 4 different bases. Adenine always base pair, thymine, cytosine always base pair 1. And the 2 strands are linked by complementary base pairing and twisted into another. So, one DNA molecule, learn it this way again. One DNA molecule is made of 2 polynucleotides. Right? Each nucleotide consists of 3 things. Sugar phosphate. There are 4 bases. Then you talk about complementary base pairing and the double. So 1, 2, 3, 4. One DNA, 2 polynucleotide chains and antiparallel. Each nucleotide consists of 3 things, right? Sugar phosphate base. Then 4 bases. And the complementary base pairing. And the 2 strands are linked. And then twisted into double. So that is the structure. So when if you go by 1, 2, 3, 4, you won't miss out. You will get your 5 marks, I think. See, 6 marks are not 5 marks. Okay, so they expect quite a lot. So you see, asked many times, explain how a transgenic organism can be produced. What are we asking you to write down? Again, how you can genetically modify bacteria to produce insulin, right? Because transgenic organisms are bacteria that are modified to produce insulin are transgenic organisms. So explain, so you've got to describe, cut the gene pace and so on.
But then for this one, we don't need to specify that it's insulin, but we just need to say that it's a gene.
You may not have to specify, but you can give the example. Okay? Yeah. Okay? Because most people will give the example. So in your example, we see all those steps, you get it. So these are the questions that came up. So, okay, so when we link back to this, the questions very difficult. So far, not very difficult. So don't always think that it must be it's not very difficult. But you really need to know your work. You will always, not always, very often, right? Questions ask you to describe relationship between DNA, genes, chromosomes. You have seen it? Okay. Right. Describe the structure of DNA. So when you ask to describe the structure of DNA, you must always talk about 1, 2, 3, 4, all the sugar, phosphate, base pairing, and so on. The rule of complementary base pairing, I think is simple. DNA is used to carry genetic code. So every DNA molecule carries a number of genes. Now each gene carries a genetic code. It's not the whole DNA carries genetic code. The DNA carries genetic code. These genetic codes are actually carried in the genes, right? And the genes carry the code for synthesis of specific polypeptides. How does your cell know what protein to produce? How does it read the code? you must understand transcription, translation, the triplet code. Okay, the DNA model, the gene is double-stranded. The code is only written on one strand called the template. How is the code written? Every 3 bases code for 1 amino acid carries. But the code is inside the nucleus. Your ribosomes are in the cytoplasm, you know. So how you send this information out? Transcription. Copy the code onto another molecule called mRNA. How to copy? Based on complementary base pairing. However, mRNA does not contain thymine. It replaces it with uracil. It's just don't have that. It's like asking me why you got 2 eyes. Now you just have 2 eyes. I don't know. Okay, so this mRNA is small enough to pass through the pores of the membrane of the nucleus. Once it goes out to the cytoplasm, where does it go? mRNA will go to the ribosomes, right? The factories. So you attach to a ribosome, and then how does the process of translation take place? The ribosome will move along the mRNA. How? Translating, okay, codon by codon, right? Okay, so every 3 bases code for 1 amino acid. So the amino acids will be linked up according to the triplet codes. Once they are linked up, they're joined by peptide bonds, you get a polypeptide. This polypeptide is a long chain, right? But a polypeptide, you know, a protein is not a long chain. A protein is folded into 3D structure. So what does a polypeptide do? Normally it will be enclosed in a vesicle, the polypeptide, and then they will be sent to the Golgi apparatus. Right? And the Golgi apparatus, this polypeptide are modified into protein. So they will be wrapped around to form a 3D structure. Okay, and then secretory vesicles will take this protein and split them out of the cell. Can you see the whole link? Digestion of proteins from amino acid, absorption sent to the cells, diffusion into the cells, all the amino acids into the cells. So what do? They are used to make new protoplasm. They are used to make proteins. How to make proteins? Oh, this is transcription, transmission, and it's linked all back to the instruction that's inside the nucleus. Okay?
Before it is being transported to the Golgi apparatus, is it protein or is it a polypeptide?
It could be a polypeptide. Yeah, or it could be a protein, but that needs further modification. Anyway, they mentioned that the Golgi apparatus modifies the protein. Or it could be the polypeptide is now, folded into the 3D structure to give it a protein. Because every protein has a specific structure, right? For example, enzymes. Okay, so if let's say there is a mistake in the code, then you get a different amino acid. Then your polypeptide is a different polypeptide. It may not be able to fold into that 3D structure. Alright? So that's why it's called mutation. This is a mutation what? Instead of producing this protein, produce some other protein like the effective hemoglobin protein.
So if you're running out, so like before you transport to the Golgi apparatus, how you say protein?
Yeah. Okay. The proteins are then transported by vesicles to the Golgi apparatus to be modified as packaged before they are released in secretory vesicles out of the cell. Any questions? Is it effective or not? This session?
Yes.
What else can we do in this session? Because this topic has relatively few questions. And all the questions are about the same type of question. You notice, right? So when you study this chapter, you know what to focus on. It's always the same type of question. When we go to another topic, the questions may be more varied. Okay? So we also have to look at the variety of questions in other topics and how to prepare ourselves when we study for other topics. Okay? How else can this section be improved? Like what else can we do? Give worksheets. You still want to do homework? Actually, don't have to give worksheets. The questions are TYS questions. You mean you want extra questions from outside the TYS? Please. Okay, that means you want a chance to actually do it in class. Instead of I thought already you forget. Okay. Okay, maybe one worksheet, do some questions. What else can I do? Yes?
Do we need to know, the genetic, the engineering thing, right? The back of like the sweet corn, the tomato,
Those are just examples to support what are various genetic modified.
the plant, like we don't need to know what the fruit is.
Don't need to know the Okay. Okay, so worksheet. What else? You must tell me now. What is it that you need? So that the session will be more effective. Don't want you to waste your 1 hour here. Huh? 1-to-1 Consultation of course is 1-to-1 or 1-to-a-few What else? Tell me. Because it's catered to, this group like you. Because from my point of view, I'm thinking that you need to know what to study. You need to know what kind of questions will come up. And you need to know how to answer some of these questions. Right? Of course, there are, if you look at other schools' paper, some of the questions are very, very difficult. Right? But I can tell you that you scan the whole TYS, you will never see very, very, very difficult questions. They are out of the syllabus. Okay? There could be some application questions. So sometimes I feel that you don't waste too much of your time, looking at other schools' paper that test things outside the syllabus and are also so extremely difficult that you will never find it in the book. Okay, so I don't want you to focus on all those things but neglect the basic stuff. Okay, so you really have to get the basic stuff and you have the technique of answering. So next time I will, let you do a few questions, at least jot down some points so that something to take home now. Okay, so that's okay, that's one thing. What else? Quiz no point quiz. Don't waste time. Don't waste time. No, Kahoot quiz all that, don't waste time. Kahoot quiz normally you're so focused on being the top scorer that you anyhow choose right? Okay, so that's so I think we'll stop here for today. Okay, thank you girls.